Thank you. It's a pleasure to introduce our next speaker, Johanna Daly from the Albert Einstein College of Medicine. Johanna's work focuses on the basic and translational research in Plasmodium species. And this afternoon we're going to hear about HIV and malaria.
Thanks very much. It's a thrill to be back and see my old colleagues and make some new ones. And the task was to try to compare malaria and TB, and I'll do the best I can. So I'll just give you a little background, sort of update on malaria and HIV. Maybe there's some comparisons with TB, which we can discuss. And then we do have a cohort study looking at the host response in Rwandan adults with malaria, half of them with HIV, half without. And so I'll be interested in some feedback on this. So there's the typical life cycle. Sporozoites are inoculated. This is where the vaccine is targeting, the current vaccine. Liver stage for 2 weeks. The problem with the liver stage is patients are asymptomatic, so we can't really study maybe in vivo the immune response. And then the stage that we're most interested in is the stage where the parasites infect red cells and rapidly divide every 48 hours and cause disease. So this is really, most of our data is going to be regarding patients during the asexual stage infection. So this parasite's very clever. It infects red cells, which seems brilliant. However, it studs the surface with parasite antigens, which seems less brilliant, but these are variant antigens, and that's really why malaria is, you don't acquire sterilizing immunity. It's sort of like HIV in that way, is that it's constantly mutating, and in fact, we really don't gain sterilizing immunity. For the last half of its life cycle, it's 48 hours, it then sticks to venules. And this is only in falciparum. The other malarias don't do that. Our idea is perhaps it's avoiding splenic clearance. So as these red cells become abnormal, as they go through the spleen, they're taken out of circulation. So really, probably our main immune defense organ is probably the spleen. And this is really the take-home message in malaria, is that there are really 3 types of malaria infection. The one that gets all the press is severe malaria, but this is only in young children in hyperendemic areas. Most folks have mild malaria, which is just really cytokine storms, flu-like symptoms. And then I think what's probably the most fascinating but least studied is the reservoir in the village of asymptomatic parasitemia. And this occurs completely in relation to age. If you are born and raised in a malaria endemic area, you can tolerate the pathogen in terms of disease. You have no symptoms at all. There seems to be a correlation with inflammation. Higher inflammation, worse outcomes. And then like TB, which has also been around forever, we have a lot of host polymorphisms that may change this relationship. So when I think about TB and malaria, I think they're both similar in the sense they're both chronic infections. People don't really sterilize TB. They end up with granulomas and latency, and that's true of malaria as well. Patients don't get sterilizing immunity. Instead, they have decreased symptoms. So this is sort of the classic sort of data that malaria immunity is age and related. So here's the age here. If we take folks who have been exposed for 8 months, they get no decrease in prevalence of malaria. However, if you lived in the endemic area for 20 months, somehow your immune system is learning to tolerate it, so we have less prevalence. And also, this is related to age. So we get this non-sterilizing immunity. It's related to exposure and to age. Now, HIV changes this, just like it does in TB, in that folks lose this adaptive immunity. So you're older, you should have no symptoms. If you have HIV, now you're going to have symptomatic malaria. So in some regions, malaria is a sentinel disease for HIV, as is TB. But there's another type of immunity or perhaps disease pathogenesis, and that's severe disease. So severe disease is these very unusual symptoms, coma, severe anemia, but you develop very quickly, if you live in a hyperendemic area, immunity to cerebral malaria. In Malawi, where I work, people develop immunity by the age of 5. And what's interesting is you develop immunity to severe disease very quickly. Perhaps after one episode of severe disease, you don't get severe disease again. And people really don't understand the mechanism of that. People are interested because maybe we could develop a severe disease vaccine. But what's interesting is this immunity to severe disease may be distinct from immunity to malaria. And I think, in fact, HIV is confirming that. HIV does not appear to increase severe disease. So if you go to places like Malawi with these HIV epidemics and you go to the adult wards, the adults are not developing severe malaria. So perhaps this tells us that CD4 count is not as critical in severe disease protection, but perhaps more involved in preventing recurrent disease or going from mild to asymptomatic. So I'm not in a room full of immunologists really going to go through this, except to say that we also have TLR responses. That's very important in malaria. Probably type 1 interferon is important. T cells are going to help generate gamma interferon, which is critical, and help develop B memory cells. And of course, innate immune response is very important. If you go to the animal model, we could probably knock out any aspect and we could find a phenotype. So if there are particular things we're interested in, we could look at that data. But I think the bottom line is antibodies alone can prevent infection. So there's studies of just humoral antibody responses, protection against malaria. And we think for sure that antibodies are involved in protection from recurring infection. So if you have a lot of antibodies to all the different antigens, you have high concentrations, it's going to decrease the chance that you'll have recurrent infection. So what do we know about HIV and malaria? I will say there was a lot of excitement in 1990, 2000, and then people became less interested because there doesn't seem to be a big impact. So a lot of these studies are older and there's not a lot. So a bunch of folks have shown that if you have HIV, you don't make as many antibodies. There is a decrease in your glutathione and antioxidant response when you have HIV, and perhaps that's important in malaria response. And there's a decrease in monocyte function. There's just a paper in PLOS ONE where they found that monocytes don't phagocytose antigens as well with HIV, sort of correlated to some of the work that Sarah does. So there's not a lot of information that we know. But if I think again about TB, I think for sure HIV increases reactivation in TB, and for sure in malaria, we move from an asymptomatic to symptomatic infections. I guess we could think about all the different commonalities, but I guess I would think about how does HIV impact our host response, and is that important in our diseases? Certainly there's probably similarities in macrophage. We know CD4 T cells are needed to clear infection, and so on. Perhaps HIV is increasing some of our inhibitory molecules. That's important both in protection from malaria and in tuberculosis. We can talk about inflammatory responses. The one thing that I really struggled with is I'm telling you that antibodies are very important in malaria, but if you look at the TB literature, there is this idea or evidence that antibodies are not protective. However, I will point you to a review by folks from Einstein. I've drinking the antibody Kool-Aid from Einstein, but they will argue that antibodies are very important in tuberculosis. In fact, there are 8 separate labs that have showed antibodies protect against infection. So perhaps this is one area of overlap in terms of HIV infection in TB and malaria. The other thing I want to talk about very briefly is that co-infection increases viral load, and we can imagine this is just because you have enhanced inflammation with malaria or TB. And there's a little more evidence that malaria antigens drive replication of HIV in T cells, and of course activates T cell activation. It's probably important clinically. I will show you some data. I don't know how much there is in TB and viral loads, but perhaps this is something we should consider. So I don't really have to tell this group very much. We have overlapping endemicities. We don't know a lot about P. vivax in HIV because that also has tremendous endemicity. P. vivax, much lower parasitemias, although more inflammation. There's 1 or 2 studies coming out. But most of the data I'm going to talk about is P. falciparum, which is endemic in these areas. So there's been a lot of modeling, and again, you can see where the interest in HIV malaria was. That was in the last decade. But basically, we have excess malaria due to HIV, again, probably shifting us from asymptomatic to mild, and then malaria responsible for HIV, perhaps by increasing viral load in one partner and enhancing transmission. So there's probably an effect both ways. So this is sort of a dream study. This is back in Lancet 2000. This is in 8 years, 7,000 visits. Basically shows if you have HIV, you're more likely to have a malaria infection. And we can see here that it's CD4 count related. Probably none of this is surprising. And in children, this has finally been published. You know, there's not a lot of studies or cohorts of children with HIV and malaria. So this is actually fairly recent data. It took this long to get the data, but as you would have guessed, HIV-positive children have more malaria, and it's related to CD4 count. So if you have a lower CD4 count percent, you have more malaria. What else have we learned? If you're interested in the topic, this was a monster meta-analysis and in Lancet Infectious Diseases. Basically, we talked about more parasites. They're going to have more anemia if you have HIV and malaria. less clear whether there's really more severe disease. A place like Malawi, where adults don't get severe disease, we really don't see it rebounding. But if you go to an area where there's not high endemicity, where adults would get it anyway, it's hard to say where they're having more severe disease, because they're going to get more severe disease anyway. So that's why that's conflicting. I would say HIV doesn't cause more severe disease. And in children, if you have HIV, you're going to have also perhaps more anemia, coma, etc. And again, this is subgroup analysis. So what's the mechanism? We don't know a lot about malaria immunity. That's why we don't really have a vaccine, or we don't really understand it. But people have looked to placental malaria. Placental malaria is an interesting model because there's a single parasite that infects the placenta. There's a single antibody, there's a single protein on the surface of the red cell from the parasite, and so we can actually look at antibody response to that single parasite, and that particular protein is called VAR2 CSA. And the basic thing to know is with every pregnancy, you develop better and better antibodies to that particular parasite. So if this particular antibody, pretty high here, if we have HIV, less, and then with each pregnancy, if I don't have HIV, this antibody goes up, I have less placental malaria, so there's a real, again, association of antibody and protection. But as you would guess here, in HIV positive, there's no gain in the VAR2 CSA antibody. So if you have HIV, you don't develop protection against placental malaria. So again, this focuses back that antibodies are probably the key for protection against disease. So we wanted to further characterize host response to malaria during HIV infection. So we did focus on the humoral responses for the reasons I mentioned. We actually looked at 824 malaria antigens using one of the Felgner arrays. We wanted to enumerate this particular type of memory cell called atypical B cells. And if you remember, these were described in HIV. They're a type of B cell, memory B cell, and they're considered exhausted. They're sort of equivalent to T cell exhaustion. And the idea is that they don't make very robust antibodies. So this was described by Susan Moore, the NIH. And in the same group, they said, why don't we look at our malaria patients? And lo and behold, they too make these exhausted or atypical memory antibodies. It's controversial what it means. The first group said they also don't make robust antibodies. Another group recently has said, in fact, their antibodies they make are completely fine. So we wanted to look at antigen response and look at this particular subset. The second thing we did is, for discovery, is did a whole blood transcriptional analysis. And what we did was subtract day 30. So in malaria, we capture them when they're sick, and then we have to have a convalescent time, so we picked day 30, really for feasibility. And we're going to look at differences in transcriptomes. I would love to have done cell-specific, but for feasibility, for first look, we're going to look at whole blood responses. So study design, we carried this out in Rwanda, which is hypoendemic. So adults do get malaria. Malawi, if they did, I'd be very worried. And basically, we brought them back every week. And again, we would collect PBMCs day 0 and day 30, and we'll subtract. And then we're going to look at antibodies just at day 0. We did this with Kathy Anastos in WEACT. And I will say, boy, these folks had very, very, very mild malaria, which is not really a clinical designation, but you can see very low parasitemia and matched. You go to some areas, if you have HIV, you have higher parasite load. Here it's quite matched, but again, I think it's hypoendemic, barely febrile. Everyone's always sick for 3 days, not even anemic. And one thing that is interesting is the HIV positive patients don't develop thrombocytopenia. So it's typical in malaria that you get thrombocytopenia. What happened is the platelets are being taken out of the circulation. And interestingly, this doesn't happen in HIV. It has been reported before. Our cohort was not particularly immunosuppressed. Median was 405. And of course, everybody's on Bactrim. So this is our take-home message, and I guess maybe we would have all predicted this, but in general, there's a reduction in breadth and magnitude of antibodies to malaria antigens. So each one of these lines going across is a malaria-specific antigen. If it's red, that means there is an antibody. We basically have a global reduction in antibody responses. We were hoping maybe there would be some interesting differences, but it's basically a global reduction. And if you look at one patient at a time, HIV patients don't have as much of a breadth. So they have less breadth to number of antigens as compared to HIV negative. And then in terms of quantity or magnitude of antibody response, so here we have the blue is HIV negative, and for each antigen, HIV positive is quite a bit less. And these basically all statistically different. It seems to be quite correlative, so it's not like it's a specific defect, it's really just a quantitative difference in magnitude and breadth related to CD4 count. So I wanted to parse out, are folks with higher CD4s more likely to make antibodies? So we looked at the number of reactive antibodies per patient, and we see these folks who have actually high CD4 counts, T cells greater than 500, actually made more antibodies. However, there's a couple of folks here who have high CD4 counts, low viral loads, who don't have the breadth and magnitude. So perhaps trying to understand the difference between these three, we can understand more about what's driving the inability to make a high response to malaria antigens. These folks here had low T cell counts and high viral loads, and they generally didn't make antibodies very well. So we did enumerate the B cell compartment, and we see in general, here's HIV negative for naive, immature, activated. Our HIV positive had more activated percent memory Bs, and in fact, they had more atypical memory Bs as well as compared to HIV negative. Again, that's what we suspected. We wanted to correlate atypical memories to number of reactive and didn't really see a correlation. There might be something there, but that wasn't the answer. Perhaps we have to do really antigen-specific memory B cells. We then looked at blood transcriptional analysis. For each patient, we subtracted day 30. The take-home message is they shared lots of responses, which I'm not surprised because clinically they respond similarly, clinically they presented similarly. Perhaps our HIV negatives were a little bit better in the inflammatory response, nitric oxide, phagocytosis, for which there's data. And again, we're going to dig out the genes, what is driving those differences in sets. So to summarize this, we have a decrease in antigen reactivity and magnitude. We have more atyps. We have a dampened transcriptional response. The last part is just to talk about viral load. I think clinically this might be important. If you have malaria, here's a whole bunch of studies, your viral load pops up, usually returns to baseline in 8 weeks. This would have importance for transmission. So we had 27 folks, mean parasitemia was 0.4, median T cells, 3 were newly diagnosed, they didn't know they had it. 10 of our 27 had undetectable viral loads, and over time they didn't seem to change. So they seemed to stay suppressed, which was one question we had. And then if we looked at the rest, the other 16, half were not on ARVs, half were prescribed. The bottom line is there was really no changes in viral load in malaria infection in Rwanda. So that's good news. We did find some mutations. 3 of the patients had drug resistance mutations, so that may explain some of the high viral loads on drugs. The final thing is there was one patient who had an increase in viral load over time. They started with 62 copies, up to 3 million, and this patient was the one that actually developed active TB. So I feel like, and I think it's known, and there's studies that show active TB really drives viral load. I think this would be interesting to study, particularly if people are on ARVs, or are we getting breakthroughs? So I think we should go back and define the prevalence of HIV and malaria. If this is a sentinel disease, then every time an adult has a positive malaria dipstick, perhaps we should also do an HIV test. Do regions with higher transmission have changes in viral load? Perhaps going back to Malawi, if patients are on ARV, are they breaking through that or not? And to summarize overall, I'd say there are multiple effects of HIV and malaria-associated immune factors. Malaria antibody breadth and magnitude generally correlates with CD4 T cells, but again, we have some that are not associated in that way. And again, are atypical B cell memories in co-infected patients different than those in malaria infection alone? So anyway, it's just sort of a broad overview. These are the folks I'll acknowledge. We had some of this work done, some of the sequencing by Nina, and worked with Rebecca on the viral load paper, Kathy Anastos in Rwanda, and the team at Einstein. So happy to take any questions.
Johanna, a lot of great things to ask questions about, but my question has to do with pregnancy and the placenta. And given that we know that from the group in Malawi as well as in Kenya, that prima gravidas and secunda gravidas have a higher parasitic burden in during during pregnancy than women who've had 3 or 4 pregnancies at the Hofbauer cell level or the placental macrophage level. Have you had a chance to look at whether or not when you make the comment that pregnant women don't make antibody, have you had the chance to look at repeated pregnancies? That would be the first question. And the second question, with my comment of having said that there's a higher placental burden, I always thought that it was due to trying to fight HIV and malaria simultaneously, and that you're overburdened by viral load with the HIV, and therefore you can't curtail the parasitic disease as well. If that's true, I mean, the moms have lived in their indigenous setting for a long time. They may be newly diagnosed with HIV because they test because they're pregnant, and so they may have new acquisition of HIV. So that may be new, but do you think of the placenta as a sequestered site and something that needs to have its own immunologic repertoire developed over time?
Well, the placenta has helped us sort out this malaria problem with diverse antigens. Only one type of malaria parasite, the one that expresses VAR2-CSA, sticks to that tissue. So we've been able to sort of correlate the antibody to that antigen, which is really the one that's key. And again, people who have HIV, don't have HIV, each pregnancy they make more antibody. So from this data, if you have HIV, you clearly don't make antibody with each pregnancy. So you're not getting this adaptive response that you're getting in HIV negative. And we're trying to parse out exactly what you're saying. How is HIV altering the antibody response? Because we do think this is the key factor. And I think the placental disease correlates maybe is good enough. I think that does represent non-pregnant women's disease. I think it still is a good model, and I think it brings us back to antibodies and looking at antibody responses and what in HIV is deranging this effect, so such that we don't get increasing antibodies.
Johanna, you didn't see an increase in virus load, but have you been able to look at cell-associated HIV RNA?
Say again?
Have you been able to look at cell-associated HIV RNA to see if there's an increase in HIV transcription back of these cells in malaria immune activation?
Right. No, we just looked at free.
We have time just for one more quick question. Actually, if I could ask a quick question, it's maybe a bit of a naive question, not being a B-cell person. The role of natural antibodies in malaria, is there a significant contribution of natural antibodies from B2 cells? And might that be playing a role at all in the HIV population, where there doesn't seem to be an increase in specific antibody, but still they don't have more severe disease?
Right. I mean, as you're getting to that, actually non-specific antibodies can bind to these proteins around the surface of the red cell and result in opsonization and clearance. And, you know, nobody's really sort of looked at that. Everyone's sort of focusing on these antibodies, but you bring up a good point. The other things we need to do with these antibodies are do they cause sterilizing immunity? Some antibodies can increase or decrease inflammation. We should look at affinity. We should probably really go in much more depth versus just saying, are they here or not? But you're right, the role of just regular antibodies, no one has looked at non-specific antibodies. Thank you.
Thank you very much.